welcome to the channel. And today our topic is erythropoietin and its role in erythropoiesis as per the person commented, and we had made a video for them. So if you have any query, you can also comment and we'll make a video for you. Let's get started. First of all, let's look at the general structure of erythropoietin. Erythropoietin is a hormone, also abbreviated as EPO, and it is a 3.4 kilodalton glycoproteins. That means it is a 30,400 daltons glycoprotein hormone, and it consists of 165 amino acids in length. And the EPO molecule is composed of 4 helices, alpha, beta, and also these 4 helices we see here. Now if you see here, this is the 3D structure of the EPO hormone, and also you can see all the amino acids arranged. Now when we look at the erythropoietin production, that where is it produced, we can see that it is produced mainly in the kidney and in the liver, and regulating erythropoiesis or erythrocytes, that is the red cell production. So here we see that when normally oxygen content is enough, and enough oxygen is present in the hemoglobin, in the RBCs, the erythropoietin is present also in normal quantity. But whenever there is hypoxia, means there is less concentration of oxygen in blood, the stimulus for this will be the stimulus for the erythropoietin production. So here you see, in the condition of hypoxia, we have a lot amount of erythropoietin released from kidney, and it is released by the pericyte cells. You will study the picture later on. It primarily acts on the erythroid precursor cell at colony forming unit erythroid stage, inhibiting the apoptosis. We'll also discuss it later on. Let's look at the image first. For example, the step 1 is that hypoxia induced the kidney. First of all, what happens is that the kidney detects that in the blood the oxygen level is whatever low, so it will start releasing by its pericyte cells, it start producing EPO. Now this EPO, these are small hormones. EPO, we have discussed its structure. So these go and they affect on the EPO dependent stage, means the stage that is undergoing in the bone marrow and all the erythroid progenitor cells. It will only act on these particular stages in the colony forming unit erythroid, on the pro-erythroblast stage, and in the basophilic erythroblast stage, and not on the earlier or later stages. It will only act on these stages. Now erythropoiesis and erythropoietin, when we see that the process of erythropoiesis that we see here, erythropoiesis is what? It is the manufacturing of RBCs. And what is the site of erythropoiesis? As we see it in the fetus, erythropoiesis are formed in the liver and spleen, and after birth erythropoiesis occurs in the red bone marrow, in the bones. So back to the text. The process of erythropoiesis in the fetal liver and adult bone marrow is regulated by the hormone erythropoietin, which is produced in the kidney at low levels under homeostatic conditions, as we have discussed, like in the condition of hypoxia. Now, role of erythropoietin. The role of erythropoietin is to control the red blood cell production. You know, there are red blood cells that are present in our body, and they consist of hemoglobin, and hemoglobin is the oxygen carrier. So they are essential, so their production is essential and is controlled by EPO. And by regulating the differentiation and proliferation of erythroid progenitor cells, means the cell that will later on form the RBCs. So they will act on the baby cells, and later on they will turn into the mature RBCs. And all this process is occurring in the bone marrow. Now, as you can see here, we are going to study the role of erythropoiesis in EPO, erythropoietin in erythropoiesis. Don't mix the terms erythropoietin and erythropoiesis. Erythropoiesis is a process, whereas erythropoietin is a hormone that we are studying. Now, as we can start here, that we have circulations. In circulation we have red blood cells, and these red blood cells, their low quantity in oxygen in the condition of hypoxia, that will be detected by the kidney, and the kidney by pericyte cells, it will start producing the EPO, that is the erythropoietin hormone. Now it will go and bind to the receptors on the cell membrane of the cell, EPO are receptors, erythropoietin receptors. And after that, step 2 is that JK2 activation occurs. This binding will activate a JK2 pathway, and after that different pathways will be activated. For example, STAT5 pathway activation occur. So we're not going into the detail. Different pathways are activated again, and the effect of this activation is that it will act as an anti-apoptosis, means it will inhibit the apoptosis. Like we know, apoptosis is the cell death, and before this process was occurring, what was happening that these RBCs were what? These were killing themselves, these were dying, these precursors were dying. But now we are inhibiting it, means we are going to proliferate this process. We are stimulating the proliferation, or proliferation means division of the erythroid progenitor cells. And as we see that in the end, what happens is that RBCs are formed. We'll discuss these steps now one by one. The hormone erythropoiesin is essential for red blood cell development. EPO binds a high affinity receptor, that is EPOR, on the surface of erythroid progenitor cells, stimulating receptor dimerization and activation of the intracellular signal transduction pathways that we have discussed, like JAK2 pathway and STAT5 pathway, and that support erythroid cell survival, their proliferation and their differentiation to become the red blood cells later on. So once again, there is a picture that there is a EPO induced receptor signaling, it comes in bind, and then this JAK2 pathway is activated, that activates STAT5 pathway, and all this phosphorylation occurs, and then EPO induced differentiation, survival and proliferation signals are sent, and EPO induced gene expression occurs. And what happens is that as a result, in the last we get red blood cells. What does EPO bind to? EPO binds to EPO receptors on the surface of bone marrow erythroid precursors, resulting in their rapid replication and maturation to form functional red blood cells. So this is our main goal, the main function. This stimulation results in a rapid rise in erythrocyte counts, means the number of red blood cells will increase. That's so obvious. A consequent rise in blood oxygen, because as RBCs contain hemoglobin, so as more hemoglobin is produced, so more oxygen will be present in body. The role of erythropoietin. Erythropoietin is the primary regulator of erythropoiesis and promotes the survival, the proliferation and differentiation of erythroid progenitor cells. We've discussed them. Now, in the erythropoietic process, EPO induces homodimerization of the EPO receptors, means the form, the structure, the shape of the receptor changes on binding, which is located on the surface of erythroid progenitor cells. And number 2, dimerization activates the receptor associated Janus kinase 2 via transphosphorylation. This is the process that is basically happening, and we say it as JAK2 pathway. And specific tyrosinase in the intracellular portion of the receptor are phosphorylated and serve as a docking site for intracellular proteins, including one of the single transducers and activators of transcription, that is STAT5. This results in activating various cascades of signal transduction. STAT5 enters the nucleus on phosphorylation. As you can see in the diagram, the STAT5 was a P group was attaching to it, means phosphorylation was occurring. It will induce the transcription of the erythroid genes, means their transcription, the process of formation, like transcription, the whole process from replication to transcription, etc., all that process. So phosphatases dephosphorylate JAK2 and downregulate the EPO receptor. And after that, erythropoietin receptor activation seems to exert its effect by inhibiting apoptosis rather than by affecting the commitment of erythroid lineage, although the mechanism by which this occurs is yet unclear. Though I've tried my best to explain you the mechanism. EPO, here's the summary of all that we have studied. That EPO is a hormone released by kidney by parasite cells when there is low oxygen concentration, and it binds to the erythropoietin receptors, initiates signals that stimulates growth, inhibits apoptosis, and induces the differentiation of erythroid progenitors. And as a result, it increases the red blood cell mass.